Hello, everyone. Welcome to our first foray away from history and into biology. We have a good show for you today. It's everything you need to know to pass, not do well, pass the global regions. Sorry, not the global regions. That would be the bioregions. Yes. Anyway, we're going to go right through it here. So pretty much let's first talk about some of the parts. The general knowledge multiple choice questions will be part A, that's 30 points. And a mixed multiple choice and short answer dealing with the application of knowledge. So far, part B has always required students to draw a line graph, which will be worth 25 points as part B. Part C will have short answer questions dealing with your ability to apply material learned in the course to real-world situations. Multiple choice and short answer pertaining to 4 New York State labs performed during the school year will be part D. So a good thing is it's going to be really worth reviewing those labs maybe just as we get closer to the bioregions. So, the state requires all answers to be recorded in such a way that they cannot be tampered with. So you got to make all answers in a permanent pen, not erasable pen. So, Science and the Living Environment. That's the official title for Unit 1. All right, that is the official title for the first unit. So observation is what is seen or measured. An inference is a conclusion based on observational evidence. A hypothesis is a prediction based on available evidence. A good hypothesis states both the cause and the effect of something. A correct hypothesis can be tested and falsified, which means proven incorrect, using an experiment. The easiest way to write a correct hypothesis is an if-then statement. For example, if I give patients this pill, they will not get sick. That's pretty much a basic one of something you can always use. I always like to use it kind of a fail-safe method. A theory is an explanation of natural events that is supported by strong evidence. Theories tie together many scientific facts, hypotheses, and laws. A misconception is that theories are things that are opinions, or are not proven. This is incorrect because of the word theory. You got to use it in a scientific context. A scientific theory is not a simple guess or conjecture and is strongly supported by evidence. So it's like the climate change theory, the theory of evolution. Every scientist pretty much agrees that evolution is a thing, but there's no kind of formula to prove it. So a controlled experiment compares the results of an experiment between 2 or more groups. An experimental group is a group being tested or receiving treatment. A control group is the normal group. It should be identical to the experimental group in every way except for one. It does not receive the new treatment. And the way you can keep it identical is through controlled variables. Placebo is a sugar pill or fake treatment being given to the control group. An independent variable is a variable being tested. For example, a new drug, a new fertilizer, etc. An if is part of the if-then hypothesis, and the independent variable is always plotted on the x-axis. A dependent variable, the if is the part, it's always the if part in the if-then hypothesis, so if is the independent variable. For example, the hypothesis we talked about before, if I give the patients this pill, the pill is going to be the IV. The DV is the dependent variable. It's a variable that is measured at the end of an experiment. The results, per se, the then part of an if-then hypothesis, so they will not get sick. The sickness of the patient will be the independent variable in the hypothesis we just referred to. So, now we turn to graphs and data tables. Data tables are always used to organize which will be plotted, what's going to be about to be plotted in a graph. The first column in the table is for the independent variable. The second column is for the dependent variable. Each column should be titled, including the units of measurement, and the data in the table must be arranged in ascending or descending order. Both the x and y axes of the graph must be labeled or titled. The labels are typically the same ones used in the data table. Once again, units of measurement must be written with the title, and the independent variable is always plotted on the x-axis. The dependent variable is always plotted on the y-axis, and the x and y axes must be numbered. These numbers must increase by a uniform increment. That is, you got to count by 1s, 2s, 5s, 10s, etc. You can't just go like 3, 6, 10, 15, whatever. Your numerical scale should take up most of the axes. Squeezing it all into the bottom of the corner of the graph is impossible to read, and no credit will be given. So you got to be aware of that. It's very sassy. The number must line up with the grid lines of the graph, not with the spaces between them, and you do not need to start numbering your axis with 0. To date, all graphs drawn on the living environment regions have been line graphs. Any student who draws a bar graph instead of a line graph will be denied credit for this part of the test. Most likely, unless they tell you draw a bar graph, which is like, never happens. The characteristics of a good experiment. Here are the 8 characteristics of a good experiment. It can be repeated the same way and get the same results. It can have large sample size or many test subjects. They're performed for longer periods of time and test only one independent variable. All characteristics of the tested groups should be the same. They are also peer-reviewed, which means examined by several scientists to determine their accuracy, the accuracy of the experiment, and they must test the hypothesis and show whether it is wrong or right. It also has to be objective. The experiment conclusion are fair and unbiased, and facts and opinions are not mixed, and also the experiment has to follow established ethical and legal standards. So anyway, our next unit now is going to be the characteristics of living things. That's Unit 2. So the most common elements in living things are in order: carbon, hydrogen, oxygen, and nitrogen. You can also remember them as CHON. Organic compounds have carbon and hydrogen. C6H12O6 is organic. H2O is not organic. Organic molecules are larger than inorganic molecules. That's another good thing to remember. Carbohydrates are sugars and starches. All carbohydrates are made from simple sugars like glucose, and they supply energy. Enzymes may break down starches and complex sugars into simple sugars. Lipids store energy, including fats, oils, and waxes. Proteins are made from amino acids, and proteins make most of the chemicals used to build and run an organism's body. So as far as your body is concerned, proteins are by far the most important part of these 3 organic molecules. The shape of the proteins determines how they fit together, and with other it's the shape of the protein that and how they fit together with other molecules that determines what the protein can do. It's all part of the kind of lock and key theory. The shape of the proteins are the amino acids. So, 4 specific jobs of the proteins are enzymes. We'll get to enzymes in a minute. But receptor molecules on the cell membrane would be the second part. These are used to receive chemical messages like hormones. Antibodies are the proteins which fight infection, and hormones are the chemical messengers. Enzymes, and we get to enzymes, are a catalyst made from protein, and catalysts affect the rates or speed of chemical reactions. The lock and key model is one type of enzyme fits one and only one type of molecule. Change the shape of the enzyme will no longer work. This is true for almost all proteins. Very high temperatures cause proteins and enzymes to lose their shape, so they no longer work properly. That's why high fevers are dangerous. pH. The pH scale measures the strengths of acids and bases. A low pH is 0 through 6 an acid. A high pH 8 through 14 is a base, and a pH of 7 is neutral, and that is water. So all living things must maintain homeostasis. That's another big thing. Homeostasis is a balanced state in an organism. Dynamic equilibrium means that the body stays balanced by taking action whenever the balance is disturbed. Like sweating when the body is too hot is another big thing. That's how things are maintained. To maintain homeostasis, organisms carry out the basic life functions: transport, nutrition, excretion, respiration, growth, synthesis, regulation, and synthesis. And then they put synthesis twice in this list. That's interesting. So maybe regulation and synthesis is kind of what you got to do all together. So you got to know these terms for just basically transport, nutrition, excretion, respiration, growth, synthesis, regulation, and synthesis are all things that you should be knowing. Metabolism is the term to use to describe all of those life processes. Failure to maintain homeostasis will result in disease or, if serious, death. Transport. We're going to go into a little bit more transport now. Transport, one of these things, methods of transport is diffusion, which is the movement of molecules from a high concentration to a low concentration. It requires no energy, also known as passive transport. The active transport requires the use of energy, usually moving molecules from a low concentration to a high concentration against the flow of diffusion. So it's pretty much the opposite of diffusion. So osmosis is the diffusion of water into or out of the cell. The water diffuses into the cell, and the cell gets larger, and it may burst. If it loses water, being put in salt water, for example, will shrivel it up. That cannot be good. If you want to maintain your cells, never a good thing. Yeah. Now we'll talk about nutrition. Autotrophs, they make their own food, while heterotrophs eat other organisms. Name a type of autotroph. Well, just look outside. If you see a plant, if you see a tree, that's an autotroph. They make their own food via photosynthesis. It's carried out by plants, algae, and blue-green bacteria. Those are all autotrophs. It takes the radiant energy of the sun and puts it in the bonds of sugar molecules. Photosynthesis occurs mostly in the chloroplast of the plant cells. Plants have stomates in their leaves that let them exchange the grasses in the photosynthesis. Guard cells open and close the stomates to keep the plants from dehydrating. Xylem and phloem carry food and water throughout the plant. Photosynthesis gives us energy. That's a common mistake. Photosynthesis only stores energy in food. That's glucose. We need respiration to get the energy out of the food. So plants also do commit or use cellular respiration. That's another common mistake there. Another common mistake is guard cells protect plants from diseases. Guard cells only protect plants from water loss. That's a pretty important thing to recognize there. Respiration is the process that takes place from sugar molecules and places it in the molecules of ATP. ATP is the energy source of all living things. Aerobic respiration requires oxygen and yields more ATP for a molecule of sugar than anaerobic, which is no oxygen respiration. When humans are forced to get energy from anaerobic respiration, we produce lactic acid that damages muscles or the burn you feel during the exercise. Oh, like I'm writing my DBQ in Global today. That's some anaerobic that's some lactic acid getting my muscles because I'm pressing so hard on the paper and writing it. It's an example of right now the TBQ, not the best. Anyway, photosynthesis and aerobic respiration are opposite reactions. They are also important in cycling oxygen, carbon, hydrogen, and water throughout the environment. Here are some common mistakes. Plants use photosynthesis, not respiration. All organisms, including plants, as I just said, use respiration to get their energy. Respiration is breathing. Breathing is not respiration. Breathing exchanges the gases needed for respiration. The simple process of inhaling and exhaling does not give you ATP. That has to be exchanged through another process. Oxygen is used to breathe. This is backwards. Breathing is used to get oxygen. Oxygen is then used to obtain energy from chemical respiration. Without oxygen, you have no ATP and no energy. All living things need oxygen / need to breathe. Anaerobic organisms do not need oxygen and do not have the need to breathe, but most they do need oxygen, living organisms. So photosynthesis and respiration, they're all processes. Photosynthesis releases oxygen, respiration takes it in and spits out CO2, so photosynthesis can come back and take it in there. That's the back and forth process we all know and love today. Anyway, here is another one. A stimulus is a change in the environment. This is all part of regulation, coordination, and control of other life functions. A stimulus is a change in the environment you respond to. A neuron is a nerve cell, and an impulse is the electric signal carried by the nerves. Neurotransmitters are chemicals that help you carry this impulse. A hormone is a chemical signal secreted by different glands in the body. Examples of hormones include insulin, adrenaline, testosterone, and estrogen, also progesterone. Sorry, I totally butchered that pronunciation. I do apologize. A hormone is a chemical signal secreted by different glands in the body. Examples of hormones include insulin. That's what I just said. I know. I'm so sorry. I'm going to move on now. My fault. Don't kill me, please. Anyway, receptor molecules are proteins on the surface of the cell membrane that receive signals from the nervous system and endocrine system. These are needed for your cells to communicate and work together. As with all proteins, it's the shape of the receptor molecule that determines its functions, in this case, which signal it may receive, how it recepts, if that's even how you say it the correct tense, but whatever. So now we turn to cells. Cells are the basic unit of life. All living things, except for viruses, are made of cells. You must know the cell theory. All living things are made of one or more cells. Cells carry out all of our organisms' life function. All cells come from other cells. You also must know that the following organelles and their functions, cell